Based upon the variable number of tandem repeats assignments, it seems there's some confusion about how to analyze that type of data set. So I thought I'd give you some additional background information just to make it very understandable and very clear. Thus far in Chapter 8, we've talked about extensions of Mendel's studies. So these are things Mendel didn't see, but that we now are able to understand, such as incomplete dominance, codominance, and pleiotropy. We then talked about sex determination and sex linkage, especially those X-linked diseases that are found only on the X chromosome. Then we moved on to talk about pedigree analysis and how to set up those family trees. And now I want to briefly highlight the process of DNA fingerprinting. What are the tools used in forensics, and then how do we actually apply those? So let's start by talking about forensic tools. So the key idea here is that people are very similar. Based upon the Human Genome Project, we know that people are somewhere between 99.5 and 99.9% identical. So with forensics, what we're doing is capitalizing on that 0.1% to 0.5% difference between individuals. We use these for things like paternity tests, solving crimes, and various medical mysteries. So where do we find these differences in people? It's mostly in the genome in regions that we call the intergenic regions, so the regions between the genes. So our genes, for the most part, are quite similar. We do have some variation and some differences, which give us our variability that you see, but we also regulate our genes differently. So the regions between our genes, which we used to call junk DNA, we now realize is very, very important. So some of the differences in the regions between the genes, and SNPs actually can be also within your genes, are called SNPs, or single nucleotide polymorphisms, and I had you watch a video from 23andMe, which explained what SNPs were. We also have various markers. So this is just simply a chromosomal location for a particular part polymorphism. Poly means many, and morphism is form. Lastly, tandem repeats. So this is a repeated sequence. This is not in a gene. It's a non-protein coding region of DNA. And you can have great variability in this region. So if there's a particular tandem repeat, say CGGTA, for one individual, you might have 5 repeats of that, and for another individual, they might have 50. It's not actually changing the gene expression, it's just changing the number of repeats between genes at a particular location on a chromosome. So here again is showing you what a SNP might look like, a single nucleotide polymorphism. This is just a single change. So rather than a CG pair, which we see in the DNA labeled as number 1, that gets converted to a TA pair. The various repeats, you can see here we have a tetranucleotide repeat. So this is a repeat with 4 base pairs, tetra meaning 4. So the repeat here is GCAT, and you can see that that is repeated 3 times. So we would say for that particular repeat, it would have it would be repeated 3 times, whereas on the bottom, that GCAT is repeated 6 times. So there's variability in the number of times you see that repeat. And we can use that, because this region is so highly variable, we can use that to identify differences in individuals. So just to make sure you're good on the nomenclature, when we use the word allele, that's just basically a form of the gene or of the marker. Genotype is the 2 alleles for any gene or marker in a genome. So for genotype, you have to remember you've received 1 copy of a particular chromosome from mom and 1 copy of a particular chromosome from dad. So you got from mom chromosome 1, a form of her chromosome 1, and from dad you got another form of his chromosome 1. And that's true for all your 23 different chromosomes. Your overall DNA profile is the summation or the accumulation of all your different genotypes. So it's all the genotypes for all the chromosome markers for that particular data set. So here what you can see is with regards to marker A on chromosome 1, this individual has 9 copies of that particular variable number of tandem repeats from one copy of the chromosome, say chromosome 1 from mom, we could say it's the one on the left, and the one from dad on the right has 4 repeats of that particular marker. So now when we look at the genotype, we would say the genotype is 4 and 9 with regards to marker A. If you look at marker B, what they received from mom was 11 copies of that particular repeat, and from dad also 11 copies of that particular repeat. So we say that they're homozygous with regards to marker B. So when we're doing different analyses done by the FBI, what they actually use is 13 different markers, 13 regions on various chromosomes that have high variability. I think in England they use somewhere between 9 and 10. So the US is considered the leader with regards to this type of testing. So where do we actually get the DNA? The DNA can come from things like your hair root, blood, skin, bone. Bone can even be partially degraded. We can also get this from various body tissues as well as body fluids. So the first step in this type of analysis is you have to amplify the DNA sample that you've obtained. And I'm not going to go into detail here. I've included links to videos on the class website, as well as your book goes into great detail on this. You can refer to chapter 8 for an explanation of PCR. I'm not going to highlight that here. The main idea is that you will undergo usually about 30 rounds of DNA amplification. Each round is doubling the amount of DNA, so you get an exponential amount of DNA produced, such that at the end of this approximately 30 rounds, you have over 1 million copies of a single piece of DNA. The 3 key steps of PCR is denaturing the DNA in step 1, breaking apart that double-stranded DNA. You then anneal your primers, and then you extend that doing with DNA polymerase. Okay, so once you've amplified the DNA that you're interested in, we can then cut that with restriction enzymes that flank the variable number of tandem repeats. So here, the repeat that we're interested in has the sequence CGA-TCGA. So for student 1, you can see that this repeat is repeated on, let's call it chromosome 1. So chromosome 1 from mom, this student has 4 copies of that repeat. And on chromosome 1 from dad, the student has 5 copies of the repeat. Whereas for student 2, on chromosome 1 from mom, for instance, they have 6 copies of this repeat, whereas on chromosome 1 from dad, they have 3 copies of the repeat. So if we were then to run this out on a gel in which you separate out the DNA based upon size, you would see that there's vast differences. For student 1, you would see that it has a certain migration pattern that's very distinct from student 2. So we run these out using something called gel electrophoresis. It will separate it out based on size. This gets transferred to some type of filter paper and then probed. So let's go ahead and analyze what this looks like. So those are our tools. So let's do some actual forensic work and see if we can figure out some mysteries here. So with regards to your particular fingerprint, each individual is going to have a specific DNA profile, which is based upon their DNA. Like I said, you will usually have to amplify your sample using PCR. This can introduce some contamination and hopefully you're degraded. Then run this out on a gel and you can analyze it. As I said, each person's set of fragments is going to be unique to them. For a particular child, all of their DNA has to come from either mom or dad. So here you can see we have a gel which is showing mother on the far left, father on the far right, and their 2 children in the middle. So if we're looking at the first child on the left, we call this child 1, You can see that that first dark band that they received, that's actually from mom. But the next band down is migrated slightly lower than what mom's second band is. So if you look to the far right, you can see that that second band actually was contributed by dad. And we can work our way down. So every band on this gel needs to either come from mom or from dad. If we look at child 2, you can see that top band came from mom. And then the next band that the child received actually came from dad, which is farther down on the gel. And you can just systematically work your way down and contribute every band on this gel to either mom or dad. So as I stated, children are going to inherit one chromosome from mom or from dad. So for any particular gene or marker, the individual has to have one allele from each of them. We can use that information to do things like paternity testing. So here we're looking at 4 different markers. We have the child shown here on the left, the mother next to the child, and then the 3 potential fathers. So for marker 1, we see that the child has 2 different forms of this particular marker. They're heterozygous with regards to this marker. So they have 21 copies of this variable number of tandem repeats from one parent, and they have 30 copies from the other parent. So if you look at mom, you can see that with regards to this marker, she has 30 copies of the variable number of tandem repeats and 36. So we can attribute the 30, because we know that that's the child's mother, is due to the mom. So now we need to find a father that has 21 copies of this variable number of tandem repeats contributed to the child. So based upon that, we can rule out father 1 because he has 31 and 34, which is not a match. At this point, we really cannot confirm whether it's father 2 or father 3 because they both have 21 copies of that particular tandem repeat at marker 1. So now let's move down to marker 2. So with regards to marker 2, the child has 4 and 10. We can attribute the 4 to the mom because the mom's possibilities are 4 and 9 for that particular tandem repeat. So we need to find a father that can contribute 10 copies of that particular VNTR to the child. And so based upon that, we can conclude that father 2 is probably the father because his possible contributions are 10 and 11. If we work our way through all the markers, we can confirm the rest. So with regards to marker 3, the child has 16 and 24. We can look and see mother contributed 24, so 16 has to come from father, and that is a match with father 2. Lastly, with regards to marker 4, it's 6 and 12. We can see that mom contributed the 12 copies of that particular tandem repeat, and again, father 2 did have 6 copies of that repeat, so that's a match. Father 1 also has 6 copies of that particular repeat, but he was not a match for the other markers. So we can conclude at this point that father 1 and father 3 are not the fathers. Father 2 looks to be the father. And usually they would analyze many more markers with regards to doing a paternity test. So again, here's another type of data set that one might see. So we have mother on the left, child next to the mother, and then father 1 versus father 2. So again, every band on this gel that the child has needs to come from either mom or dad. So I'll give you a second to see if you can figure out who's the father. Is it father 1 or father 2? So hopefully you concluded that father 2 is the dad. You can see based upon that second major band in the child that is a nice match with father 2, but not with father 1. And if you track your way down the whole gel, you can see that father 2 is a nice match. We can also use this type of analysis to do various crime scenes. So what we have here are several suspects as well as evidence taken from the scene of a crime. So lane 3 is showing evidence here, and that was a vaginal swab taken from the victim. And then in lane 6, what we have is a semen stain that was taken from the victim's clothing. So what we're trying to do at this point is determine if suspect 1 or suspect 2 matches more closely the evidence taken from the scene of the crime. And what you can see is that, especially if you're looking at the evidence 1 as well as the suspect 2, there's a very strong match in terms of basically the semen that was taken from the victim from the vaginal swab. So it appears that suspect 2 is indeed the criminal in this case. Another type of analysis you might see is another application. Here we have the blood taken from the defendant and the blood taken from the victim. And blood was identified on the defendant's body, who was arguing that he was not the murderer. However, blood was taken off of his clothes. He has his jeans, and blood was found on the shirt, and especially the blood found on the shirt of the defendant matches nicely with the victim's blood, putting the defendant at the scene of the crime. This type of analysis has been fundamental in lots of different cases, one of which was trying to help bring closure for the families who had members of their families at the World Trade Center, but yet were not able to 100% conclude that their family member had died at the scene of this tragic event. So subsequently, some 1,700 people of the nearly 3,000 that were at the scene of the World Trade Center were ultimately identified using these types of forensic tools, bringing great closure for these families.